Well, hello. Welcome to a new series that we are undertaking, the first in our series of laboratory management that saves lives. And our topic today will be the CAP guidelines that have been proposed for some years now, having to do with analytical validation for immunohistochemistry. So our intent with this is to talk about the things that you can do in your laboratory to make sure that the patient results you're providing really do what they're intended to do and can provide some degree of predictability as to response and certainty of diagnosis. So the guidelines were developed with the intent to ensure that each assay was validated prior to reporting on patient samples. Obviously, that's important so that we can preserve patient safety. In the process of talking about this, however, I think hopefully will help you understand a little bit about the manner in which guidelines are developed and how and what evidence is used to refine them. You'll recognize when revalidation may be needed or required, and we'll talk a little bit about fixative and specimen type and handling some of those pre-analytical variables that can affect things. So in terms of standards, I think we would agree that a laboratory should be or is required to, depending on the legal domain in which you're operating, should be required to validate all assays and reporting systems before testing and reporting patient specimens. This is a widely held laboratory performance standard. Now, typically, we apply this just to the standards that are, or the technical aspects of things. But as this very nice poster that one of my colleagues put together a few years back demonstrated, it can also be applied to how pathologists themselves interact with various reporting systems. In this case, demonstrating when a new reporting system for lung cancer came forward, how that would be reported and how we would validate whether or not that was worthwhile. Well, Archives of Pathology thought our work was worthwhile. I'm not sure anybody has cited it since it was published, but at least conceptually, you might think of your pathologists just like you think about any other analytical system in the laboratory. They're a part of that process, and the standards that they use for diagnosis, the standards they use for reporting, if they're part of a system, certainly should be brought within the framework of things you think about validating. Well, in terms of immunohistochemistry, there is a problem here. We know from various proficiency reports, various feedback systems, that there's quite a bit of variation, both in the performance and, of course, in validation. There are currently consensus guidelines for HER2 and estrogen and progesterone receptors on breast specimens. And there have been efforts to refine the guidelines for HER2 testing in other organ systems. But we now have an expanding menu of predictive and companion diagnostic assays that are entering use and which we will need to attend to if we're going to provide reliable results and truly predictive information. So this guideline published in 2013 by the CAP and again in Archives was an attempt to define the principles that were involved and to survey the results of various laboratories. This was based on some data from prior surveys that seemed to indicate there were some problems out there that might explain why the variability in some of these assays and the results could be so drastic. For example, only 2/3 of laboratories had written procedures for validation. Only just over half specified the number of and types of cases that needed to be included in the validation study. and less than half could identify when the procedure indicated revalidation, and really only a third addressed cytology specimens. So this is a good evidence that a fair number of people out there were struggling with this issue. When various different types of things were involved, such as antigen retrieval and so forth, a fair number, two-thirds to three-quarters of laboratories had some guideline, but there was still a significant number who had not. So with that background, the CAP decided to convene an expert and advisory panel to review the published data and to develop evidence-based recommendations to the degree that there was evidence, and consensus or expert recommendations to the degree that they could offer that. Now, this was not the first time this has been employed. In fact, there are a number of guidelines. The most widely used is that from the Institute of Medicine, which says that these practice guidelines must be developed in a transparent environment, that conflicts of interest needs to be managed, that the panelists need to be from a variety of backgrounds. In general, if they're going to advocate for treatment, they need to have a patient advocate, and there has to be systematic review and considered judgment in the final development. In the case of the CAP panel, the chair was Dr. Fitzgibbons, and here you see the names of the expert panel members, the advisory members from administration, and the staff supporting it, both technically and procedurally from the CAP. So the component of systematic evidence review involves identifying the key questions, doing the literature search and data extraction steps, and then from that, developing some proposed recommendations. This is followed by a very important open comment period when the proposed recommendations are published and the public and users, patients, others are invited to comment on each and all of the recommendations before they're implemented or endorsed and published for promulgated use in a widespread process, which would follow after a considered judgment of both the comments and the data in support of those judgments. So the key questions that were involved for the CAP is what is needed to validate an assay initially before any immunohistochemical test is put into use, and when should revalidation occur, and what would be the requirements based on the change or upheaval that might have occurred. This was important to assess the analytical sensitivity and specificity of these tests, accuracy, and precision. These questions, of course, we think of often in the clinical laboratory, but we also need to apply these in the anatomic pathology laboratory as we begin to do more of these sorts of things. And so the scope of assay was, scope of evaluation included these questions. Another question is that what is the minimum number of positive and negative cases that would be needed to initially validate an immunohistochemical assay for its intended use. And in this regard, there was some consideration given to the different types of purposes to which these assays are directed, specifically those that are primarily diagnostic and non-predictive, those that are predictive or that imply a particular treatment, those that may be involved with identification of organisms, and maybe some rare antigens. And then a sort of a sub-question here was, does expression level of the antigen make a difference in terms of the number of cases that need to be evaluated, and should or how should those kinds of variable expression levels be included in the diagnostic, in the validation set? We know that those cases are included in the proficiency level examinations that are given, at least on the clinical lab, and to a degree certainly in immunohistochemistry as well. They also wanted to consider various pre-analytical variables like fixative, decalcification, time, fixation or decalcification, and any other validation that might be required when tissues have been processed in an unknown manner, say in another laboratory. And then, of course, what conditions mandate reevaluation. So the literature search component went back almost a decade. It included nearly 1,500 studies. These were reviewed by the panel, from which 126 studies were identified that merited full data extraction. Then the evidence was evaluated based on quality as well as the quantity and consistency of the evidence. Now, this quality assessment was graded according to different criteria. This is contributions from a methodology consultant that looked at the quality and execution of the studies, how many cases, how diverse the study was, and how consistent and generalizable that evidence was. Did it include adequate descriptions? Were those the basis for the right answer? Were these test results reproducible? Was there any evidence of bias or other sorts of things? And was the data analyzed properly? Based on this, the strength of evidence was graded into 3 categories: convincing, adequate, or inadequate. And this involved classifying the study in terms of its level. So level 1 studies were the best studies, and those were those that included a large panel, well-characterized samples, and had summary data from external proficiency testing schemes or from interlaboratory comparisons. So this is a very public, open standard with well-characterized standards. Level 2 would be high quality but peer-reviewed studies, but maybe perhaps not with the same level of validation. Level 3 data was either lower quality or non-reviewed studies, or they were expert panel FDA summaries, for example. And then, of course, level 4 is unpublished or non-peer-reviewed data. So if you landed a 1 or 2 level study, had a reasonable, consistent results and generalizable, that was convincing evidence. Step down for that was maybe some not totally generalizable, or maybe not quite as diverse and rich a number of cases. And inadequate were those where there were unexplained inconsistency, or there were 1 or more lower quality studies, or only expert opinion was involved. And then based on these, the strength of recommendations are made along with each of the recommendations. The strongest recommendations go where there's convincing, consistent, generalizable, good quality evidence, and further studies are unlikely to change the conclusions. If it's just a recommendation, the strength of evidence is adequate based on limitations, but further studies may be needed and may change the conclusions. And then finally, we have sort of an expert consensus opinion. This is an important element, but the strength of evidence is usually not there. And there may be reason for making the recommendation, but there are still gaps in knowledge that may change the recommendation or provide refinement. So once the systematic review is completed, the open comment period ensued beginning in July 2013 of 18 draft recommendations and 5 questions. And from that, they developed over 1,000 comments from 263 respondents. Here we see how, for their various recommendations, how certain of them merited very strong agreement. As you can see, it's likely these are going to be easily accepted with these 90, 100%. But some of them may be a little bit lower, 65, 55, 70. Some of these may have been subject to then further refinement and considered judgment as they took in that feedback. So this considered judgment process includes the feedback, the quality of the evidence. This is the point at which we look at risks and benefits, the benefits and harms, value and costs that might be associated with implementing a certain standard. looking at any regulatory requirements, what various experts have espoused. And from that, the 18 recommendations were whittled down to 14 final recommendations in this particular situation. Now, we know that this process can be iterative, and experience with the ASCO HER2 guideline is a nice example of this. So the recommendations from the initial guideline published in 2007 was that 25 to 100 samples ought to be approved or ought to be studied to perform the validation. However, in 2013, this was reduced significantly with the current guidelines based on FDA-approved assays and only 80 samples for laboratory-developed tests. However, this may have been a moot point if the assay was previously validated under this guideline and had performed successfully on proficiency testing subsequently. So that was a nice way out in that regard and didn't require revalidation. And that's important because we recognize that validation does represent a cost burden that's placed on the laboratory, and often this is not a remunerated cost for many laboratories. Similarly, the concordance required shifted between guidelines as well. So for any category, any cases that were less than 95%, that had to be reflexed to some alternate method. However, in 2013, those concordance requirements were changed. The laboratories only had to comply with accreditation and PT, and any specific numeric requirement was lifted. So there is obviously evidence that use of guidelines over time will provide guidance as to what ones are helpful, what ones are an overly heavy burden, and what seems to be the best thing to go with. So looking on the bright side, not paying much attention to that bee hiding in one of the florets there, let's go on to what the guidelines are. So for immunohistochemical testing, laboratories must validate all immunohistochemical tests before placing them into clinical service. This is a recommendation, and in laboratories that are accredited by the CAP, that is an operative requirement. So they can use a variety of things to validate the tests. Correlating a new test with the morphology and expected results could be acceptable. Comparing that new test with something that's been validated in the same laboratory previously, that's acceptable. Or comparing with a validation set validated in another laboratory that has a validated assay would also be acceptable. The strength of evidence for this guideline was fairly strong. It was adequate to support when validation should be done, but it was not adequate to assess how the validation should be done. So we recognize that there is some variation. We know that analytic validation provides a net benefit for the overall performance, but there is some measure of flexibility here in terms of how the assay is validated and what may be the optimal way to avoid potential harms that might be associated with analytical false positives or negatives. This clearly is what we're trying to avoid, so that is the key thing to be thinking about is where am I going to use my skills to avoid these two things right here. That's what we don't want to get to. And with certain assays, that's more of a consideration than with others. So the validation issues that are involved here. What do we put in the validation set? What cases do we choose to validate? Well, in general, we'd like to see positive, negatives, and low positive tissues. So we'd like to be able to be pretty clear that near those threshold cutoff values that we're going to still provide consistent results. Some of these should be diseased tissues. They should not all be normal tissues. And as much as possible, they should reflect the intended use of the assay. Now, as a clue here that you don't need to have one slide per assay, clearly a particular tissue sample that includes both positive and negative cell types on the same section could be utilized and thus drastically reduce the amount of reagent or tissue that needs to be assayed, particularly in the situation where we'll talk later where TMAs may be employed for some of these sorts of things. So for the guideline number 2, for those assays that are used clinically, laboratories should achieve at least 90% overall concordance between the new test and the comparator test. If the concordance is less than 90%, laboratories need to investigate the cause of low concordance. So this doesn't mean that automatically if you fall less, you need to repeat the whole study. You have to uncover the cause. So repeat testing, did the assay not work on that sample? Does a higher antibody titer make a difference? Was the tissue fixed differently? Or is there some other variable that you haven't accounted for that has caused you to drop below that 90% threshold? For this guideline, the strength of evidence was quite adequate to support the 90% as opposed to the 95%, which had been the previous threshold with the ER-PR-HER2 standard, and was certainly an overall adequate for analytical validation. And here's some of the data. I won't go into the particulars of this in terms of the reported studies that showed this kind of data. So one of the interactive portions of our discussion here is at least a self-assessment. Here you go. You're working in a laboratory. Does your lab have a procedure or policy to validate the performance of new immunohistochemical antibodies before they're used and reported? And if you don't know, if your answer is unsure, may I suggest that you go to talk with your laboratory director and say, where can I find this procedure? And if they say, oh, we don't have one, well, you may be able to raise your hand to say, can I write that? I know where the guidelines are. Now, the second question is, does your validation procedure distinguish between predictive and non-predictive assays? So here's the clue. So predictive markers are standalone tests that provides information on a likely response to a given therapy. So an example, rituximab. What's the CD20? What's the CD117? Is this patient going to respond to imatinib? HER2 is another example, of course. Non-predictive markers are usually done as part of a panel and interpreted only in the context of other morphologic and clinical data. So that's the importance here. This is one that says up, down, yes, no, drug, no drug. These figure into a component of a panel, supporting a diagnosis, refuting a diagnosis, et cetera, et cetera. All right, third question. Do you know how many cases, positive and negative, your lab normally would use to validate a non-predictive marker, for example, HMB45? And this would, of course, be not excluding rare antigens like, ALK or Whipple-I or something like that. Is it less than 2, 3 to 5, 6 to 10, 11 to 15, 16 to 20, more than 20? What's your answer? Well, while you think about that or go look to try to find that out, let me introduce guideline number 3, excuse me. This gives us the numbers. For initial analytic assays for non-predictive assays, laboratories should test a minimum of 10 positive and 10 negative tissues. So a total of 20 cases, 10 positive, 10 negative. Now, the laboratory medical director has some flexibility here. And if he determines that less than 20 is sufficient, for example, if it's a rare antigen and finding 20 positives would take you from now until doomsday, then you need to document the reason for that. And in general, your assay set should include high and low expressors of positive cases if that's possible. And usually you would want to make sure that you're spanning the range of clinical results. So if you're going to use this on colon and endometrial cancers, well, then you should include both tumor types in your assay group. And if you're going to report quantitatively, say a Ki-67 or something like that, then you need to include a variety of expression levels. So how much evidence do we have for this standard? Well, it's inadequate to support the number of validation samples. So we're going to admit that. And that's why the medical director has some leeway to exert his judgment in cases where they may not be able to readily acquire 20 positive and 20 negative. But it is adequate to support the distinction between predictive and non-predictive markers. So we want to do that. Now, just if we think about this, this gives you some idea of the math involved. And I'm not a statistician, I'll admit that. But if we look at the number of cases, this will give you an idea of perhaps why we have chosen this standard. So if we just have 10 samples and there's none of them that are discordant, our confidence limits are still pretty broad, see 70 to 100%. If we have one discordant cases, then it drops down to 50. And if we have two discordant, we're under 50. So that's really not a very high confidence level, high confidence interval. On the other hand, if we have 20 cases and we have only one discordant case, well, that gives us 95% concordance with a confidence interval of 75 to 100%. And if we have two discordant cases, which would still be acceptable, we're still nearly 70%. So we're still quite favorable in this confidence interval. And so that's the reason why this number is 20 rather than 15 or 5 or some other number. So let's change gears and talk about a predictive marker. So how many cases does your lab normally use to validate a predictive marker? Is it less than 5, 5 to 10, 10 to 15, 16 to 20, 20 to 40, or more than 40? Well, you're waiting to hear the number, I'm sure. Guideline 4, for initial analytic validation of all laboratory-developed predictive marker assays, laboratories should test a minimum of 20 positive and 20 positive and 20 negative tissues. When the director determines that fewer than 40 is sufficient, the rationale needs to be documented. And again, this expected range of clinical results and use cases should be included in what is assayed in that validation set. So how strong is the evidence here? Well, again, we don't have solid literature to support the recommended number of validation samples. Having said that, I think this has been in operation now for several years, and there have not been any significant studies that have refuted this study to my knowledge. Now, you can correct me if I'm wrong, and I will freely acknowledge that, but to my knowledge, that has not been superseded. It is, of course, as we mentioned, sufficient to support distinguishing between predictive and non-predictive markers. So what does the data look like numerically in this situation? So obviously with 40 markers, you can have up to 4 discordant markers and still have, up to 4 discordant tests in the validation versus the test set and still pass the assay. And that gives you a confidence interval still greater than 75%. So that's pretty good math. Not optimal, but it's certainly pretty good to indicate that the highest likelihood that you're giving a good result, even with 4 discordant tests, is pretty high. As opposed to with 20 cases, you lose that even with 2. You're less than 70%. So that's the rationale statistically for these numbers. And we'll show you here a sample validation study. So here's a 2x2 table of 40 tissue validation set that did not meet the benchmark. So instead of having 4, it had 5. 5 results that were outliers. And that meant that the concordance was 87.5%, and that this would fail. Now, can you do anything with that? Well, you can certainly reinspect these and see if there's a reason why they may have failed, why you may have turned negative instead of positive. Maybe you had exhausted the tissue. Maybe there was some other pre-analytic or analytic issue with those samples that might shift that and therefore salvage your assay without repeating it on a whole new set. Okay, so we've exposed the bees, and hopefully you haven't felt stung by this standard, but it is good to know where these guidelines are going to take you in terms of the confidence intervals with your test. You want to be able to say, I have confidence in this test when your clinicians question you about the result or something that doesn't seem to fit. But knowing these numbers, you of course recognize that any biological system has some areas for gradation. So guideline 5, a marker with both predictive and non-predictive applications, you should use and validate it as a predictive marker. The evidence is certainly adequate to support the use of higher validation standards in that situation, and so there's no big gaffe about that one. Guideline 6, when possible, laboratories should use validation tissues that have been processed and use the same fixative and processing methods as cases that will be tested clinically. Well, obviously, we know that pre-analytical variables are one of the big issues in immunohistochemistry staining, and that was a potential pitfall for prognostic assays in the breast and required some measure of standardization as to fixation times. And so there's some evidence to support that wherever possible, these similar methods should be used and applied. We also know from circumstances where other processing methods like microwave processing and so forth have been employed that this will be an indication or a need for potentially revalidating, retitrating antibodies and so forth to ensure that performance is optimized under the new regimen of handling and processing. This is a potential burden. We recognize this for reference laboratories that receive tissues with varying fixation protocols. We see this obviously in staining and with other staining methods where local recipes may differ, and certainly have seen this in internationally processed or submitted samples that chemical fixatives, quality of fixation, and so forth can be quite different in different settings. So these have to be interpreted very carefully if they're going to be used. And ideally, if someone is going to use you as a reference laboratory for this service, then you should consider attempting to, or making sure that their samples are included in your validation study in sufficient numbers to not destroy your confidence in the assay. So one question arises, what do you do with cytology specimens? Has your laboratory validated immunohistochemical assays for cytology? Well, I'd be surprised if any laboratory is answering yes for all markers. If they are able to answer yes for some markers, that's good. And if they've answered no, then they may not have thought about the question deeply enough and may need to. guideline here is only an expert consensus opinion. In other words, there's not a lot of data out there, but the consensus expert opinion is that if you're going to do immunohistochemistry regularly on cytologic samples, that they should be, and they're not processed in the same manner as tissues that are used for your assay validation. In other words, if they're prefixed in alcohol or otherwise handled, they're smears or air dried or other sorts of things, then you need to consider using those cases in part of your validation set to be able to confidently state that your assay is going to work for those samples. And that certainly would apply for predictive as well as non-predictive markers. So the strength of evidence here is really not great. We can't address the criteria and the number of samples needed for validation with cytology. And in general, if you haven't fully validated those, you may want to include a disclaimer in your report to indicate that immunohistochemistry on cytology specimens is subject to cautious interpretation due to this validation issue. So here's another question. What about decalcified specimens? Well, we are certainly aware that decalcified specimens come into the laboratory with some regularity. Oftentimes we get a bone sample with metastatic carcinoma, and the question is, where did it come from? What kind of tumor is it? What are the markers? Is this, can this be treated? And so consideration should be given to validating decalcified samples, especially for some predictive markers where that request may come frequently, like HER2. Again, the decalcification methodology should be considered. So acid decalcification versus EDTA decalcification may provide some preservation of antigens that would otherwise be destroyed or degraded through prolonged acid-based decalcification. Okay, we'll take a breath here for a second and let's go on then to guideline number 8. Again, this is an expert consensus opinion. If immunohistochemistry is regularly done on decalcified tissues, laboratories should test a sufficient number of such tissues to ensure that assays consistently achieve expected results. And the medical director has discretion in determining the number of positive and negatives that will be done on predictive or non-predictive markers to test. Again, where's your level of comfort with and confidence in your diagnosis? Obviously, if you get a sample of 5 that's all perfectly concordant, you may feel more confident. But if you get 10 and you have one discordant, you're still okay, but in fact, your confidence interval may have been accelerated or may have been accentuated and improved with that result. Again, the strength of evidence for this is inadequate to identify the criteria numbered, but focused validation on representative samples that are used frequently with decalcified specimens, keratins, various site of origin markers, PAX8, CDX2, TTF1, those sorts of things may be very worth employing in a standardized assay to validate for decalcified specimens. And of course, this doesn't have to be a specimen that actually has bone in it. It can be just a tissue sample that is subjected to your decalcification processing procedure. So guideline 9, here's one of the great helps in this process, and that is that tissue microarrays or multi-tissue blocks can be used in the validation sets as appropriate. Whole sessions should be used if there are circumstances where tissue microarrays may not be appropriate, such as those where you have variable, patchy expression of the assay, and you need to have a greater volume of tumor to assess appropriate expression. Similarly, you want to make sure that any of those tissue microarrays or multi-tissue blocks have been handled in the same way. So don't put a Boolean fixed sample into your multi-tissue block and expect it to react in the same way as your formalin fixed sample. Again, the guideline here is adequate to support TMA usage. These are very widely used. but the variables involved with each marker need to be considered. And the other things to be wary of, as I've mentioned, when heterogeneity is involved, such as gastric HER2, or when there's very limited areas of tissue expression, such as BCL6 and other markers. Well, okay, so are you ready for the next section? Here's a question to lead into that. How often do you completely revalidate an immunohistochemistry assay when the antibody is changed to a different clone? So this is a pretty significant change in your procedure. Should you do it always, usually, occasionally, never, or you don't know? Well, while you're going off to look at that, here's some Indian paintbrush that's a little bit on the pale side from up high in the mountains. So as we look into the next several guidelines in terms of revalidation, the strength of evidence for these is essentially not strong. It's inadequate to assess exactly which conditions require assay revalidation. So we're looking here at recommendations based on expert opinion and a fairly minimal amount of evidence. With that said, there is some measure of adjusting the stringency under which revalidation is required. So these may be, in the case of antibody-specific issues, fairly modest in the case of just a new antibody lot, or maybe a little bit more stringent when you're changing the dilution or the same clone from a different vendor, or you're changing some aspect of incubation or retrieval. But when you're changing clones, that's when it really matters. And for all the assays, certain other issues may warrant a level of revalidation that will help to reinforce that the change has not significantly impacted your process results. And that can include fixation, antigen detection system, tissue processing equipment, moving from paraffin to, moving from liquid fixatives to microwave fixation, or even environmental locations, different humidities, different water supplies. Those warrant some measure of revalidation. So guideline 10, as I've mentioned, expert opinion. When a new reagent lot is placed into clinical service for an existing validated assay, they should confirm the assay's performance with at least one known positive and one known negative. That's a pretty low bar and should be readily achievable for most laboratories. You may want to include low expressors, especially if that's a predictive marker that you're using. Guideline 11. Laboratories should confirm assay performance with at least two known positive and two known negative. So here we've stepped it up a little bit when we're changing dilution, vendor, or incubation or retrieval times. And again, low-level expressors may be good to include in that situation. So this is a little bit stronger, but still not a very impossible bar to clear. Guideline 12. Laboratories should confirm assay performance by testing a sufficient number of cases to ensure that assays consistently achieve expected results when any of the following have happened. Fixation type, antigen retrieval method, in other words, going from pH to heat or something of that sort. The antigen detection system has changed, tissue processing has changed, or you're moving to a new equipment vendor, or when other laboratory conditions such as water supply or location are changed, this should be done. Now, this is when the director is responsible to determine how many predictive and non-predictive markers should be tested on this. A reasonable approach would be to select a variety of antibodies from your menu that have variable clinical uses, both predictive and non-predictive, have different antigen localization. So some membranous, some cytoplasmic, some nuclear, et cetera, some matrix, whatever, and variable antibody types, monoclonal or polyclonal. So that gives you an idea of being able to revalidate your assays in those circumstances in a reasoned manner to inform confidence in interpreting these results in a variety of antibody types and situations. And then guideline 13, again, expert opinion, laboratories should run a full revalidation when the antibody clone is changed for an existing validated assay. So that's the most significant change you can make in your assay function, and revalidating entirely with a new clone is what's involved. So guideline 14, laboratories should document all validations and verifications in compliance with regulatory and accreditation requirements. If you don't have it written down, if you don't have the documentation, it didn't happen. That is the voice of the inspector speaking. And I've been there, so having a good record-keeping system for both procedures and validation studies is very, very important to your laboratory. Okay, so with that basis, let's take a few minutes and do some problems. So these problems are oriented around predictive markers, change of clone, new diagnostic marker without a reference test, and then a predictive marker where there's a change in assay conditions, okay? So just sort of test yourself to see how well you captured what I've talked about here. So here's EGFR, okay? We've done 12 samples here. And as you look at this, you can see, oh, there may be a few things that are discordant here, okay? Sample 4, sample 6, sample 10, and the discordance goes both ways. So is this assay validated? Well, in theory here, EGFR is going to be a targeted therapy. First off, we didn't use enough samples. Second off, we don't have 90% concordance. So the answer is no, we are not yet validated with that assay. What further needs to be done? Are you thinking? Okay, more samples. That would help to strengthen your case. Evaluation of your discordance to look for explanations or issues, or further sampling till you get 90% concordance. Those would all be suitable steps. If the medical director looks at those and finds there's an adequate explanation for those discordant assays, they can, in their discretionary judgment as a director, make that choice to deem the assay validated. Okay, case 2. Oh, we got a new clone of S100. Oh, golly. We had that old clone, it worked so well, but now we've got this new one. So here's our validation study. We did 7 cases. And what did we get? Well, we've got 1 discrepant. So is this enough? Are we validated? Remember, it's a new clone. So it's not well validated. And what needs to be done? Well, for a new assay of a non-predictive marker, 20 cases, 10 positive, 10 negative, should be done, unless the medical director deems otherwise, and a 90% concordance rate should be achieved. So we need 13 more cases in a suitable mix of positive and negatives. We've got 4 negatives and 3 positives in our sample group, and so we just need to bolster those numbers and get to the point where we can say we've done 20, and then look and see where we are in this process. Okay, you're getting the idea here. All right, new antibody to D240, and you have no experience with this antibody. You've never used it before, and none of your peer laboratories in the neighborhood are using it. You're the first guy on the block with this test, and you do your assay and you go, oh, we've done 20 cases. That's our standard number. And look what we've got. We've got 2 discrepants. Well, is it validated? Yes, we hit the bar. Now, here's a trick question. What is the fewest number of slides that could be used for this validation? Well, we mentioned, of course, that tissue microarrays can be used, or multi-tissue blocks. So you conceivably could have a tissue microarray with 20 tissue samples that were all suitable to be tested within the target realm for this antibody. And so you could theoretically do this with one slide. Now, should more than one time of running the procedure be used to validate reproducibility? I think that's a question in your individual laboratory. Depends on how similar this assay is to other assays you're running, and how frequently you'll be running it. But you may make that decision. It's not a requirement. But assessing inter-observer, inter-run reproducibility is a component that should be considered in this testing as well. Okay, well, here's another case for. So let's say we've got a change in chromogen for our estrogen receptor predictive marker. Which of the following is sufficient to validate this protocol change? So, 2 positives, 2 known negatives with just the new platform. 10 positives, 10 negatives on the new platform. 20 positive, 20 negative on the new platform. Or 2 positive, 2 negs on both the old and the new chromogen platforms. Well, in theory, A is a suitable and acceptable change. Acceptable choice in this situation. D would also, of course, be acceptable, but you're not really changing the hormone. You're just changing chromogen here. But what happens if one of the positives doesn't stain? Well, then in that situation, you're going to have to go on and do more in order to hit your standard idea, or you're going to have to troubleshoot it and figure out why one of those didn't stain. Is there something else? Is there chimeric, steric hindrance or some other process going on that is making this new chromogen less reliable than your previous one? So you have to evaluate and look at these and investigate them to see and find the problems. Well, if you're like me as a laboratory director, these are sometimes a headache. And many laboratories are doing these time and time again. Large laboratories may add 10s, 20s, 30 new markers every year as new things become available, and some of them will be predictive and some of them will be non-predictive. So for many laboratories, finding sufficient cases is the big issue. This is particularly an issue with rare antigens, infrequently used antibodies. There is work out there working to develop quantitative expression in tissue surrogates. This is still in the developmental stage. It has promise. I don't think, if there are products on there, they're not widely on the market. They're not widely used in our laboratory. But in general, this is usually where the problems are. And along with that, of course, staff time, expense involved. This is where you can go to your vendors and say, I need this much for my validation study, and you should be able to get that. For some larger laboratories, validating alternate specimens is also an issue. Well, I'm going to skip this one. So as we think about what we've talked here and the way in which we're trying to make these assays really sing for us, really be something we can pound our fist on the table and say, yes, this is going to predict the response to treatment in this case. We want to make sure that the results we're giving are true, that we're able to identify this flower very reliably because of its immunohistochemical profile. Well, there is evidence out there that the publication of these kinds of standards and guidelines is helpful. This study conducted a few years following the publication of the guidelines just looked at how many now had procedures. And you see, whereas before we were at 65, 75, now there's a fairly large number, of vast majority, 85%, who have written procedures specifying the numbers of cases, and also specify minimum numbers for predictive markers as well. So, at least on this one criterion, and there were others in the study that were also included that showed similar progress. Our colleagues in the laboratory director world have made progress. So our summary take-home messages, our colleagues in the clinical realm, they rely on accurate diagnostic and prognostic testing. If we're going to give them an answer, they need to bank on it. And analytical validation is critical to ensuring that this is going to perform as expected. Using guidelines can be an important way to achieve reproducibility and consistency, as well as feeling like you are in the mainstream. But this also underscores the fact that lab management is a team activity. Both the directors as well as the technical staff are involved. But we also need to collaborate with each other, especially as we look to bring more of these things into other areas where currently they're not available. Because certainly one of the hardest things in a developing area is to validate results, and having an appropriate number of samples to do that validation and appropriate guideline on which to work is very valuable. I want to express appreciation to the CAP Quality Center, aka the Center, specifically Drs. Wagar, Fitzgibbons, and Goldsmith, who helped to develop a lot of the content in this presentation, and to Lisa Fothery, also at the CAP, for her help. I hope you've enjoyed it and appreciate you joining me. We intend to do further sessions in lab management, so please subscribe so you'll catch on to those. And if it was useful, I'd appreciate your feedback and comments. Other topics in lab management you'd like to see covered, likewise, please include those in the comments, and let us know what we can do to make your life and your work helpful. So until next time, thanks for joining us.